Je prends mes yeux.
Ça passe comme ça.
Normalement, ce couple, il marche pour toute la salle.
Ok, ouais, donc on peut se mettre là et ça va. Je pense qu'on commence à... Ouais, c'est bien, ça va avoir une idée. Ça se comprend par rapport à ça prend 3 ans, tout ça. J'aime bien ce truc où toute la idée, et voilà.
On a fait un peu rapidement.
Ouais.
Tu fais à ouvert. Par contre, tu peux passer les dias, ouais.
2 minutes. Ouais, voilà. On commence. Ok. Okay, so we're going to start. So it's my pleasure to welcome you to this first conference in the memory of Isabelle Olivieri. And so we thought that first we need to give you a little background about why we created this new conference. And because I see here like new faces, we thought also it would be good to tell you about Isabelle a bit. Personally, I didn't know her, but I think she was very important for the community. And so I will try to kind of line up a few stuff about her background, basically. And so why we created this event is that maybe you know, maybe not all of you know, but we have a second conference later in June called the Louis Thaler Conference that happens every year, and this for a while. And we thought it would be nice to have another conference earlier on at the beginning of the year in September. And because to kind of balance the sex ratio, we thought that it would be nice to name it after a female researcher because it's important that they are also represented at this type of event. And so that's the reason why we named it after Isabelle Olivieri. And now I'm trying to explain you why we think that Isabelle is inspiring enough so that we named this event after her. I think it's important here we've been discussing about role model. What's a role model in science for students or for early career researcher? The idea is that for students, it's not that you will become Isabelle or you should become Isabelle. It's more that it's someone that is inspiring. Of course, she was human like all of us, and she was not perfect. So the idea is not that it's a perfect image and that we need to reach this kind of ideal image. It's more about really inspiring and having these people that did a lot for the community, that helped really to structure the community and to know about their work and just to be inspired and maybe sometimes to give you confidence in your career, because sometimes when you're a student or early career researchers, you feel that you're not good enough. And it's just to show you that some people, they achieved a lot and they help other people to grow. And it's more with this idea that we named this event after her. And so I will just give a few background about Isabelle. So she passed away a few years ago, like 7 years ago. And first, so it's really, her career to me is really interesting because she was first a researcher at INRA until the mid-19th. And then she studied a professorship here at the University of Montpellier. And she was the first professor in evolution and in population genetics in Montpellier. The first female professor, right? Because there were male professors before that. Her main research themes were dispersion, metapopulation, speciation, demography, and mixing demography and genetics, but she had really a lot of interest. I won't go through all her diplomas and awards. I will just maybe mention the CNRS Silver Medal that she earned in 2007, and the Grand Prize from the French Society of Ecology and Evolution. Back then it was French Society for Ecology that she earned in 2012. She did a lot to teach ecology and evolution in Montpellier. She started a first-year bachelor program in theoretical biology, and it was really good to kind of attract math students, actually, to bring them to biology. She also studied the RPI master, and now it's integrated into the BE master. Sorry about this, I don't know how to get rid of it. But yeah, anyway. And then in 2010, she started the Erasmus Mundus master program, which is joint with other universities in Europe. So Groningen in the Netherlands, Uppsala, and Munich in Germany. So that's for the kind of the background. And then maybe more about like some other things and the diversity of things Isabelle have done. First off, I think she was like a great professor. Personally, she was not a professor for me, but everyone I've been talking to, they were really emphasizing how good of a teacher she was and how good she was to kind of communicate complicated concepts in, for example, in population genetics to the students. She was an inspiring researcher. She made a lot of links between theoretical approaches and empirical approaches. She did do some experimental work, mostly at the beginning, from what I understood, like at the beginning of the experiment. She made also links between basic and applied research, because remember she was working at INRA at the beginning of her career. And I think she really contributed to kind of structure the research community of Montpellier. And so I feel that now we kind of inherit from this, from these years, basically. She was also committed at the international level in various, sitting on various committees, for example, just to mention some for example, at the ISEM, the European Society for Evolution Barrier, and also at the EMBO. And I think this also contributed to kind of increase the visibility of our community at the international level. So that was, I think, something very good for us. And with this, I will let Jeanne introduce the next topic.
So beyond this curriculum, we also wanted to let you know, especially for those of you who didn't get the chance to meet her, a bit about who she was. And to do so, what we did is that we asked people who knew her to send us a few lines about how she influenced their research careers, but also the discipline in general. And so sorry if not everyone could contribute to this. We did that a bit last minute. But so now we are just going to read just what we received from people in the community. And sorry, only one of this is in English, so we will just briefly skip to French. So the first one was, I will always remember a generosity and welcome, a huge reason that I'm still in science now.
Second one is, Isabelle m'a appris qu'il faut avoir le courage de se jeter dans l'inconnu et de garder la liberté d'esprit.
Isabelle aimait rassembler les gens et les idées et faire des communautés où elle évoluait une grande famille. Elle m'a appris qu'on pouvait réussir en sciences en cherchant avant tout à y trouver de la joie, sans peur et avec générosité. Elle était aussi un modèle de liberté.
Isabelle m'a appris à toujours essayer d'aller au bout d'un argument en évaluant toutes les alternatives possibles. Elle adorait argumenter et avait comme habitude, pour pousser au maximum les arguments de l'interlocuteur, d'adopter des positions auxquelles elle ne croyait pas nécessairement. Elle a participé avec d'autres au développement de la biologie évolutive à Montpellier, en France et en Europe. Et elle a particulièrement œuvré pour l'intégration des considérations écologiques en génétique des populations et réciproquement.
Tisseuse de liens qui libèrent.
Isabelle avait le plus beau carnet d'adresses de la biologie évolutive en Europe.
Elle me fait un peu penser à une sorcière magnifique. Son labo était son chaudron dans lequel elle faisait des mélanges sans suivre de recettes.
Ah, c'est plus long. Oops, sorry about that. Okay. That's this one, right? Isabelle a aussi été très importante pour la communauté de chercheurs, d'enseignants et d'étudiants qui travaillent sur les questions de diversité des plantes cultivées et leurs organismes associés, symbiotes, parasites ou pathogènes. Elle a eu là, comme ailleurs, un rôle essentiel comme chercheuse à l'INRA par ses travaux et aussi par sa capacité à faire venir auprès d'elle de jeunes thésards et chercheurs, et enfin comme enseignante dans le master Poétiforme d'EPS puis RPI en lien avec l'agro de Montpellier. Son impact a vraiment été très fort. Son énergie, sa gentillesse, son humour, son franc-parler étaient tellement géniaux que c'était toujours un privilège de passer un moment avec elle.
C'était une professeure extraordinaire. J'ai assisté à pas mal de ses cours et il y en a eu plusieurs où je me souviens en être sortie en me disant que c'était dommage que ça n'ait pas été filmé. Quand elle était en forme, elle arrivait à captiver les étudiants en rendant les cours interactifs sur des sujets compliqués, tout en restant décontractée et sans être surplombante. Quoi qu'en aient dit certains, au moins à une époque, et même si elle avait son franc-parler, elle avait aussi beaucoup d'empathie pour les étudiants qu'elle aidait bien au-delà du cercle restreint des étudiants de son labo. Elle était aussi une chercheuse hyper vive, curieuse et intéressante sur à peu près n'importe quel sujet, avec un énorme réseau de collaboration qui a beaucoup profité à la communauté locale.
Tu m'as dire, inutile de me demander ce que je ferais dans 10 ans. Si je le savais, je serais déjà en train de le faire. Et moi, 10 ans après, je me surprends encore à travailler sur des questions que tu m'avais inspirées. Je ne te remercierai jamais assez.
One is a bit special. Isabelle, c'est la retranscription anecdotique de ses premiers cours en M1, ex-maîtrise par les étudiants. À quoi sert le mal? À faire le café. à vider le marc de café, à réchauffer le café, à rien. Il existe maintenant des machines à café.
Il n'y a pas la recherche fondamentale d'un côté et la recherche appliquée de l'autre. Il y a juste de la bonne et de la mauvaise recherche.
Isabelle a contribué à m'apprendre à se jeter à l'eau sans savoir nager. Avec elle, rien n'était impossible, enfin presque. Elle a toujours refusé qu'on mesure des flux de pollen par fluorescence en se baladant sur les falaises de la classe. Okay, so with that, we just wanted to have a few thanks to acknowledgements. Ça passe ou pas? Okay, I will remember. So the first thank was to Fadela Tamoun, who contributed to the organization of this meeting. Then we wanted to thank the PhD organization committee, which always brings nice activities to the speaker and the PhD students. So Gwen Vigo, Mathieu Hull, Tanguy Muller. Also, Isabelle Barbeau for this nice poster. And finally, all of the contributors of the testimony we just read. And with that,
well, I'm really delighted to have the privilege to introduce the first speaker of the first, the speaker of the first Isabelle Olivieri conference, Frédérique Viard. Basically, when the organizing committee was considering a local figure for this first Olivieri conference, Fred's name came up almost immediately. Fred is a research director at CNRS. As you probably know, she's back in Montpellier for 3 years now. Actually, Fred is a true Montpellieran. From her birth to her PhD at ISEM in 1996. Then she dispersed away from Montpellier for a postdoc at UBC in Vancouver. Then her first position at the CNRS in Lille and a move to the Roscoff Biological Station in 2000. There, she worked on various marine species and ecosystems for 20 years before returning back home for our great pleasure indeed. Fred's research is a sublime example of the ecology and evolution interdisciplinary approach. It focuses mainly on dispersal processes and more specifically on anthropogenic dispersal, the study of introduction routes, dispersal patterns, the evolution of non-indigenous species and their impacts. She's somehow the queen of marine invasion. And for all her work on marine invasion, she was awarded the CNRS Silver Medal in 2018. Fred is also a member of some national and international expert groups working on non-indigenous species and was the lead author of the IPBES Biodiversity Assessment in 2018. So thank you, Frédérique, to have agreed to give this first conference. And now the floor is yours.
Can you hear me? Well, it's difficult to take the floor after all these, the first and the last part. Well, thank you for the kind words, and thank you for also the presentation you did before. It was really emotional, and it's truly a pleasure to open this new cycle of conference dedicated to Isabelle, who has been, of course, so inspiring to me, both as a researcher and a human person, and particularly as also a woman scientist. And it's my pleasure, but it's also quite stressful. So, let's go. So, at the request of the organizer, I will cover a topic from quite a broad perspective, not too technical. It will not be a not too technical talk, I think. And it's focusing on my research. And you have it with this title, DNA Chronicles of Marine Biological Introductions in Ports, the three keywords defining my research. So as Nicolas said, the focus on marine biological introductions, the approach I mostly use, which is DNA-based studies, and the places where I work the most, which are ports. So, marine species introduction. How many marine species have been introduced? Approximately 3,000 have been reported so far, which is approximately 1.5% of all marine species that have been described so far. So the proportion is quite similar to what is known in other systems, other environments. But we know these numbers are strongly underestimated and actually are not that meaningful. What is meaningful is the trend. And you have here on the left side, a picture showing the number of marine species that have been introduced in European seas over time. And you can see this sharp increase starting in the 1970s. And this increase is due to trade globalization, especially shipping. And you have on the left a thick picture showing the same trend of a sharp increase in shipping activities over time, starting again in the 1970s. And this parallelism between these two trends is simply explained by the fact that shipping is the major introduction vector of marine species. Introduction vectors in marine system are diverse, and you have here 14 different categories of vectors that have been reported by Jim Carlton, and they are very, very diverse. Education, research, leisure activities like with scuba diving, of course, all the offshore platforms, of course, fisheries, aquaculture movements, etc. But shipping is responsible for more than half of this introduction at a worldwide level, and it's also true downscaling at more regional level. And all kinds of ships can actually transport species, either in their ballast, so these big tanks, which are used to balance the ship during its journey, but also on the hulls and including on big cargoes. You have here the diversity of ships contributing to this marine species introduction. You have military boats, you have research boats, you have, of course, all the cargoes, passenger ferries, fishing boats, and leisure boats, which for a long time had been particularly neglected as a vector, but which now appear as to be a major introduction vector and also playing a major role in spreading species introduced in a new area. So shipping is building this kind of network at global, regional, local scale. And you have here, for the global scale, a picture showing you the travel of 30,000 ships, the largest one. And what you can see is that they are really breaking natural biogeographic barriers. And globally, but also at a regional level. For example, here on the right, you can see a picture showing, the travel of 600 boats from different marinas spread out the Mediterranean Sea. And many of these boats are crossing the Mediterranean Sea from east to west and vice versa. And also some of them are creating a connection, a link between the Mediterranean Sea and the Atlantic coast. And the number of these vectors is also quite massive. Just to give you two numbers, for the big ships, commercial ships, 1,000 ships are traveling in the world, and they are responsible for 80% of the global trade volume. And for leisure boating, in France only, we have more than 300,000 leisure boats which are registered. So it's really numerous. And of course, ports are the nodes of these networks. And they are the nodes at a global scale. On the left, you have a picture showing the distribution of commercial ports according to their size. And on the right, with the marinas, again, quite widespread at a global scale, with areas like on the eastern coast of US and also in Europe, you have more density, higher density of marinas. And again, the numbers are quite impressive. For example, in France only, there are 462 maritime ports. And they are both increasing, actually. They are increasing, for example, for marinas by almost 20% over the period between 2013 and 2018. So this is increasing, and this is contributing to what has been called blue acceleration and the increased artificialization of the cost. So, through shipping, these ports, which are the nodes of the network I showed you, are sources and recipients of marine introduced species. I show you an example with a survey, an inventory that was made in southern Western Atlantic, and particularly the coast of Argentina, where you have here the number of marine species that have been reported, and on the right, you can see where those introduced species are established, and most of them are found in ports. So there is a tight link between these ports habitat, and the introduction of marine species and their establishment. So it's obvious that ports are natural playground for studying marine biological introduction processes. And this will be the aim of this talk today by addressing 3 questions and showing you this connection. The first question I will address is, are ports biological communities singular? And in particular, what is the non-native species contribution to this biological community? You will see a lot NNS in my slide. It stands for non-native species, introduced species. The second question, are ports and natural habitats connected? And it's linked to the question of a spread of non-native species from the place they were originally introduced towards other habitats. And the third question, are ports a melting pot for anthropogenic hybridization? And so it's dealing with the issue of non-native species impacts. So let's start with the first question. Are ports biological communities singular? And so the underlying question is to determine the extent of non-native species contribution to port communities, so to quantify or assess what is named the colonization pressure, the number of species which are arriving. To quantify this colonization pressure, the primary task, of course, is to be able to identify those species, which is quite challenging when using traditional approach, which are based on morphological characteristics, because many of these marine introduced species are belonging to complex taxonomy groups, like in hydrozoans, bryozoans, tunicates, for which, unfortunately, the taxonomic expertise is strongly declining. Also, by using this method, you need a lot of resources in terms of people involved, in terms of time needed, and you have to examine every specimen one by one. Of course, these methods are not effective when you are dealing with cryptic species, which have been defined based on molecular traits only, and which are quite abundant in marine species introduced. And also not always effective for early stages, at least in some groups, so for juveniles or larvae. Thus, thanks to recent advances in sequencing technique, there has been a shift towards using this molecular approach that has been named metabarcoding approach, which is presumably an effective approach to circumvent the limitations that I just mentioned. And so I guess that many of you know this approach, but basically, my pointer is not working, but basically, you take samples either from water imports or scrapping the hull of the ship. You collect all the specimens organisms, extract all the DNA, go through high throughput sequencing, a set, and it's only one line, but it's a lot of work to determine the good tool, bioinformatic analysis, and from the set of molecular sequences you get by using reference database, making the correspondence between some particular sequence and some particular species, you can get theoretically a species list. So this is a theory. In practice, it's a bit more complicated than that. But this is the idea, and I would like to show you an example with this case study, which is a study of, that was carried out as part of the PhD of Marjorie when I was in Roscoff. And I take this opportunity to thank all the Roscoff team who was really helpful for this work in, on marine species imports. And in the study, what we did is that we sampled specimens scrapped under pontoons, and we sampled at the same time water samples to make both a traditional an inventory of the species present. So with the species specimens collected with quadrats, and at the same time metabarcoding, and we targeted 4 taxonomic classes. Gastropoda, bivalvia, Acidiacea, and 1 class of bryozoans in these 10 ports, and repeated that over 2 seasons. I summarize briefly the results with this graph showing you for each port and the 2 seasons, fall and spring, and by the metabarcoding and the quadrat survey approaches, the proportion of taxa in pink non-native species and in green, blue native species. And as you can see, first, the results are quite congruent between the 2 approaches, but more importantly is that this sessile introduced animal within these 4 taxonomic classes are represented by a substantial fraction of non-native species, whatever the season and whatever the port. And it can reach up to 40% of the species present in a given classes. So there is really a large contribution on non-native species as compared to native species to the biological community for these sessile animals in these classes. And so when we compare now the composition, the biological composition of the community of the species assemblage in marina as compared to natural habitats, we have this kind of picture. In this picture, the color refers to the port in orange, and the other points are natural subtidal rocky habitats, which are favorable habitats for the species that we were examining in the port. And you have here results from PCA, obtained from morphological data and metabarcoding survey. And as you can see, both also clearly show that the marina are clearly separated from the natural habitats. So this shows, whatever the approach, that there is really a port singularity, which is explained mostly, or partly, I would say, partly by this high non-native species contribution to the established community. So we have really high colonization pressure, high number of non-native species, but which is at the same time coupled with high propagal pressure, a high number of introduced individuals. The propagal pressure is a combination of the size of, for a given introduction event, of the inoculum, so number of specimens of a given species introduced at what time, and the wave introduction, number of introduction events, both make propagal pressure. And for this first component, it's obvious that in marine species, it's easy to introduce a lot of specimens because there are numerous microscopic stages like larvae, spores, there are species with high fecundity, etc. And regarding the introduction events, there are also numerous introduction vectors. I mentioned for ships, for example, the diversity of vectors, and many of them are unregulated. So it creates perfect conditions to have a high propagule pressure. And several theoretical and empirical, as well as meta-analysis, showed that propagule pressure is a major contributor of non-native species establishment success. For example, because it decreases the effect of environmental stochasticity, algae effect, inbreeding and inbreeding depression, etc. And another aspect of this high propagule pressure is that the introduced population are highly genetically diverse. I show you just one example with one solitary tunicate, Stilina clava, that you can see on this picture here. This is a big tunicate, which actually here is wrapped. You see the creamy layer? This is a colonial introduced tunicate, which really like to settle on the solitary one. Anyway, so for this species, which is native to Asia and has been introduced in both North America and Europe, if you look at the average genetic diversity of the introduced population and compare this average genetic diversity with the average of the native population, you actually see that this is all similar. And for certain populations, the value is higher than the average in native populations. And this is a result which is not uncommon. Actually, this is almost the rule. By examining results of genetic studies for 59 marine introduced species in Europe, we could show that this pattern was observed in 76% of these marine introduced species. So most of the time, we have this kind of scenario in which there are numerous specimens introduced from different sources that contribute to the genetic diversity of population in the introduction range. And of course, this can be seen as a fuel for selection on large standing genetic variation or on novel genotypes that will rise through admixture, recombination and admixture. Regarding this first question, the take-home message and the answer is simply yes, ports are singular marine habitats. Of course, they are first artificial habitats that are replacing or fragmenting natural ones. They are well known to ports display numerous environmental specificities from an abiotic point of view, like sun pollution, light pollution, artificial substrate, contaminant, etc. But this is also important. There are also important hotspots for marine non-native species establishment. And as I show you with the contribution, and the melting pot reshuffling the diversity both at taxonomic, at species level, and within species of the genetic diversity. So reshuffling the diversity at various scale and through shifting. So now I come to the second question, how ports and natural habitats connect. And so behind this question, the aim is to trace the non-native species escapees and also to analyze and understand better the non-native species population connectivity patterns. I will jump directly into one example to address this question with this beautiful seaweed. Well, I think it's beautiful. they introduced the Pacific kelp Undaria pinnatifida that you can see here in the port. It has a nice story, interesting story in terms of introduction. It's native to Asia, and it has been first accidentally introduced in Europe, in France, in the Tour Lagoon in 1971. Accidental introduction likely to be connected to the deliberate introduction of the Pacific oysters, Magallana gigas. But because this is an edible seaweed, you know it. It's the wakame, which is used in the soup. It's an edible seaweed with high added value and commercial value. So it was decided to transport it in Brittany, to cultivate it in three pilot farms, with these crazy ideas that the species would not be able to reproduce in the cold Brittany water. But the species is in its natural range is living up to Vladivostok. But apparently, there was evidence that it could be safely cultivated. Of course, the story which follows was expected. There were escapees from farms the year after the culture was set up. And so it became an introduced an invasive species in Northern Europe. If looking at the situation in Brittany, nowadays it's like this, it's really widespread, and especially it is found with one exception to my knowledge, but in every marina of the coast of Brittany. It's also still cultivated, at the start here, and it can be found on natural habitats, particularly in some bays like here in the Gulf of St. Malo. And it's not surprising because it's really a species that like living in marinas, and the colonization of marinas can be very, very fast. We had the opportunity to survey the colonization of a newly built marina, and we did these surveys over 3 years of this newly built marina, geolocalizing all the individuals. And you have here the picture over time, February 2014, March, December, and then for the other years, the subsequent year. And you can see how it spread, and it's really a snowball effect, classical snowball effect of this colonization. Interestingly, doing genetic analysis, we could show that the first individual found on the infrastructure itself were for 20% originating for individuals that we collected the year before on hull boats. So there is again this link between shipping and establishment in artificial habitats. This link is interesting because this is a seaweed with very limited natural dispersal ability. The spores are living a few hours. There are no big, like, you know, in mollusk where you have larvae living 1 month in the water column. It's not that kind of thing. They are very, very poor natural dispersal ability, but it's easily transported on the hull of ship. You have here an example on a leisure boat when you can see a nice sporophyte developing and attached. And there are also all the gametophytes, the microscopic stage, which can also settle on different structure and be transported. So what we were interested in by using this model was to determine to which extent the populations in marinas exchange genes with population established in natural habitats. I will not address this question, but we have in mind their specific adaptation or something like that. What are the connectivity gene flow between population established in this network of marinas and ports? So during his postdoc, Charo used a genotyping procedure based on DD-RAD sequencing to analyze roughly 800 individuals, which were sampled within different bays along the coast of Brittany. And for each bay, we wanted to have at least one pair of marina triangle and natural population in a natural habitat, the cycle. And within 2 bays, here and here, we also had the cultivated population. I will summarize the result by showing you these plots. This is it's not structure, but kind of structural analysis. So all the individuals are ranked along this line, and there is 1 line per individual. And they are sorting according to the location of sampling. So here, this is the first marina, here, La Trinité. Then you have all the individuals from Quiberon. Then you have a marina and a rocky reef from Etel, etc. And this, from the south to the north along the coastline, and here the 2 farms. And so the color shows a membership to different genetic clusters that are best explaining the overall genetic variance in the data set. And so what you can easily observe is that each pair of marina and rocky habitats, which are close, located in the same bay, are very, very similar. So within a bay, natural habitats and birds population share a common or genetic background, or similar genetic background, and it's very clear and it's very distinctive from the farms. So this, together with observation from the field, was supporting this idea of spillover from marinas, but not from farms. And so there is clearly a change in the source of natural population over time. I told you the story of the pilot farms at Enscape. So at the very beginning in the 1970s, both marina and wild populations were coming from the farms. I put a dotted line here because actually marinas were first colonized and very little wild habitats at the beginning. And then there had been a spillover from marinas to the wild, and the farms are now very little contributing to the marina and the wild, which is expected because they are not very numerous, and they are not exerting a strong pressure, demographic pressure. The second, using the same graph, we can see something else very different. This is the fact that sometimes populations which are very close together are very dissimilar, but populations located far away together are conversely very similar. And this is the classical pattern observed in many marine introduced species living in the sports and on hard substrates, that there is this mosaic genetic structure which is supporting the effect of long-distance dispersal events due to leisure boating. And really this kind of structure and mosaic genetic structure are formed quite commonly for short dispersals. And it's true for, of course, introduced species, but also for native species living in these habitats. And it has been shown, for example, on a native tunicate species, Siona intestinalis, where you have, again, which is a very short dispersal with larvae living only a few hours. And again, you have this total lack of correspondence between genetic distance and either the geographic distance or the current distance. So, for the second question, the answer is that yes, ports and natural habitats can exchange migrants, and it can have spillover of non-native species from ports to the wild, as well as spillback. I'm not sure yet which use that, but in the colonization of this newly built marina, we could see that some natural habitats also contributed to the colonization of the marina. But very important findings of all of this different genetic work is that also shipping between ports provide connectivity shortcuts to short dispersers. And so we have this mosaic genetic structure at regional scale. This kind of exchange between natural and port habitat also raised many questions regarding the spread of specific adaptations that would evolve in ports and what would be their fate in natural habitats, and in particular for native species, when native species are colonizing both natural and port environment. And this is something which is still very little studied so far, and it needs to be investigated precisely. So now I come to the last question. Are ports a melting pot for anthropogenic hybridization? So we know that shipping, by breaking natural biogeographic barriers, lead to secondary contacts between distinct lineages and distinct species that were previously in allopatry. I often mention, for example, non-native species coming to Asia and now found in Europe. And from these secondary contact occurring in ports, there are diverse possible outcomes, like coexistence of the two species, or competitive interaction up to competitive exclusion, but also when the two species are not reproductively isolated, the possibility of hybridization, and in that case of the effect of human activities on this secondary contact. This is often called anthropogenic hybridization, leading to hybridization, and then possibly to integration. And for this question, I will show you an example with a secondary contact between two tunicate species, two solitary tunicate species. Cyona intestinalis, which is a native species to Europe. Its distribution range is here. It has been reported, it is an invasive species here in North America. It has been reported also here, but it has never been really confirmed. And Cyona robusta, native to Asia, and which has been introduced in many places around the world, but also including in Europe. And the two species are very, and so the contact zone is notably centered around the English Channel, and the map should be below up to the Bay of Biscay. And this is a recent contact. The introduction of Cyona robusta is estimated to be at the end of the 1990s or early 2000s. Interestingly, the two species are very highly divergent. 15% molecular divergence, 3 million year divergence time. So it's really species that are highly divergent. And so at first you could expect to see coexistence, competitive exclusion, but that this should be maybe reproductively isolated, but no, because despite their high divergence, they display a strong hybridization potential. First, they are coexisting in sympatry. These maps are showing you the relative proportion of the two species over 3 different years. In pink, the introduced species, and in blue, the native one. They have synchronous life cycle, but exactly synchronous at 2 generations per year, and spawning at the same time, and settlement at the same time. So ideal condition for hybridization, especially because actually they are not reproductively isolated. And F1 hybrids can be obtained in the lab, but in one direction only. The eggs are from the native species, and sperms, the male function, from the introduced species. So, strong hybridization potential, but a first study that was done during the PhD, of Sarah Boschus, that you could see on the previous slide. I forgot to mention her at this slide. But during her PhD, what Sarah did is to analyze 300 SNPs, which were selected to be ancestry informative, so really diagnostic of the two species, And you have this genetic, and they were analyzed in a set of population from the contact zone, as well in places where the two species are not coexisting together. And the results are briefly summarized with this PCA, showing you actually that all the Sayona intestinalis individuals are on the well isolated on the first axis, as compared to all the individuals from Sayona robusta on the second axis, whatever the location, so in the St. Patrick area or the Allopatrick range. There is one exception here, which is one single individual from one population located here. And actually, we know that it's a F1 hybrid. And we have a control here with the experimental F1 hybrids produced in the lab. But altogether, and you don't see anything else in the between. And so we can conclude that there is no gene flow, no contemporary hybridization between the two species. So this was my conclusion in 2017. We came to this conclusion, well, the two species are found together. Here it's robusta, here it's intestinalis. So we have this outcome of coexistence between the two species. But a different picture arose thanks to a strong increase in marker density. And this is the work of Alan and Christelle. We used Alan rod sequencing and Christelle full genome resequencing data to reanalyze this pattern. And what they found was really, really interesting. It was like finding a needle in a haystack. They discovered an integration breakthrough where in a very localized region of a single chromosome, chromosome 5, here, or alleles from the introduced species within the genome of intestinalis. So a very localized integration found only in the contact zone, and you have here a map showing the relative proportion of Sayona robusta allele genes and Sayona intestinalis alleles. You can see that there are places where it's poorly frequent and others where it's more frequent. On top of that, Christelle showed that it's an adaptive integration because they have different signatures of selection on these alleles. And there is also, interestingly, here a gene coding for a family of cytochrome P450, which is involved in the detoxification process. And the two species are living imports, so it makes sense. And this gene is located at the core of the integration region. And this is a copy number variation, so a structural variant. You have here two SNPs on this cytochrome P450. And for the two, you can see that for Sayona robusta, you have a large number of copy. And the same large number is found in Sayona intestinalis, so native individuals that had been introgressed, but not in the individuals that are not introgressed. So this is an adaptive integration involving probably copy number variation. But all the story I just told you is based on populations that were sampled in single year, 2012. And so first. And second, as I briefly mentioned, it's sometimes a very low frequency, it's variable, and it's never fixed in a population. So the question what could be the fate of its integration from starting in 2012? Is it going to fix because it's strongly adaptive and confer a strong advantage, or could it be finally lost because of too much genetic incompatibilities between the two genomes of these two species that are highly divergent. So to address this question, the idea was to make a comparison by resampling the exact same population plus additional one and make a comparison 20 generations apart. And this is the work that Fanny is currently finishing, presenting in her thesis. And so what Fanny did is she compares the relative proportion of Sayona robusta alleles and Sayona intestinalis in this integration block, comparing sampling in 2012 and sampling in 2021, which is for the two species, again, 2 generations per year, so 20 generations. And what is clear from this analysis is that the integration is very stable over time in space, also, with those regions where the integration was important, also it was the same in 2012. Where it's absent, it's the same. So it's stable over time, but still not fixed, which is questioning the processes behind, and notably considering this environmental heterogeneity, this heterogeneity, spatial heterogeneity in the patterns that may be linked to some kind of local adaptation. And from this study, we have now a new interpretation of the whole story, is that there is an anthropogenic hybridization followed by an integration, local integration. To be honest, I did not explain everything because I don't want to spoil what Fanny will present you during your defense next December, the 8th. So please to listen to her, and she will explain the whole story, and probably better than I do. And my final remark on this case study is that I really like this case study because actually it's still an evolving situation. And at the beginning, for starting this third part of my talk, I mentioned 3 different outcomes: coexistence, competitive exclusion, and anthropogenic hybridization. And actually, we are at a stage here where the species are still coexisting together. There are places where you can find the 2 species. But Sayona robusta seem to decline a bit. It's more difficult to find it than in the past. Now the native species seem to be, in many places, far more important than the introduced one. And we have the introduction where these alien alleles are found into the genome of the native species. So it's not only coexisting scenarios, there are also scenarios that can simultaneously coexist. So to finish on this third part, so yes, ports are melting pots for hybridization. Hybridization can have very, very not exclusive outcomes from hybridism, homogeneous admixture to local integration. And the best case studies, I did not present that today, but are all the Mytilus species that are studied by Nicolas. To exemplify this diversity of outcome. Integration by the introduced species into the native species genome can confer advantage, like I show you with these 2 species of Sayona. And altogether, this gives support to one of the scenarios linking colonization pressure, propagate pressure, and non-native success, is the importance of the recombination to create novel genotypes that may be advantageous in the new environment colonized by the marine introduced species. And I think it's almost here. It's okay, fine. Some concluding remarks, only 2 slides. I hope that I convince you that ports are very interesting habitats to study, but also singular ones in which non-native species play a major role. They are singular because of their particular abiotic properties, which are exerting strong selective pressure, and singular biotic feature, which is important non-native species contribution. They are forming also this network of interconnected habitat, even at long distance, providing connectivity shortcuts to even to short dispersers, and not isolated from wider habitats that may facilitate the spread of non-native species outside this singular habitat. And there is a series of evolutionary outcomes. I mentioned some of them today. The genetic diversity shuffling, the mosaic genetic structures, the admixture and hybridization processes. So they are really perfect arena for an urban rendezvous, with species and genetic diversity being constantly reshuffled. And it's a playground for studying ecological and evolutionary processes, and that's it. I would like to thank you for your attention, and I will be pleased to answer your question if any.
Whether the effect of increased genetic diversity that we've seen for the exotic species, but also observed for native species.
That's a very, very good question, because there are no study of that, almost none. You mean when you compare natural imports, I guess, natural habitats imports. Actually, the comparisons of population established in natural habitats and ports are really focused so far on marine introduced species, because this was why people start to working in ports. They did not care about the native species. But I don't think this is the case for Mytilus. You know that, Nicola? No, I don't think. So I try to think to some example. On patella, yes. From what I remember from one study, and it's really very rare studies made on a gastropod, I don't think neither that was shown. It was more, if I remember correctly, lower genetic diversity, maybe due to some kind of founder effect, because it's opportunistic colonization as compared to the natural habitats. So this increase that we observed, I agree that we need to compare that with native species living in the same habitats to determine if this is a link to some particular processes in these habitats, or simply due to this mixing. But we should see that on native species if the scenario of the effect of shipping with connectivity shortcuts would exchange would favor the exchange between populations. We should have something which is actually quite diverse locally. So like we see at the community level, actually what we see is usually a sustained alpha diversity, but a very low beta diversity. So I would expect something quite similar.
This trading integration in this particular sector. And do you have any dates the integration?
Yes, it was done by Christelle. Well, you know, this kind of inferences are often quite questionable, but it's recent. The estimation she did was around 70 years, 75 years. But so, well, but there is, of course, but it's contemporary. We are not looking at some dimensions. And again, it was not found outside the contact zone. It's only in the contact zone.
Sure to attend the defense of your PhD students, so I have a question. So in the last example you showed, so you said that between over 10 years,
20 generations ago,
decrease of the invasive species, and more or less suggested that this integration could be.
The integration itself at the core of the integration, or really central part of the integration, is stable in frequency and distribution in space.
Yeah.
So, but there is a decrease, apparent decrease, but we know from previous surveys that Sayona robusta is kind of like this boom and bust, and we don't have survey annually over the whole period to determine if this is only in 2021 that species seem to disappear or decline, or is it just part of this boom and bust? So it's to answer you.
The decrease is all over the sites that you study. Could be surprised if there was.
It has been observed in the past. In the past, before Sarah started her PhD in 2013, there was surveys were done by a colleague from US who regularly came over the same range, and she observed a steep decline in 2009 and thought that the species would have been declining and disappearing. But actually, when we started the job, it was a boof. So we guess that there are some reservoirs, not necessarily new introductions, but maybe. But we cannot exclude that. But it's a bit too, we don't have enough data to see, to really say, well, it's declining, disappearing, and what we observe is a legacy. I would like that because I like the idea of the legacy, but but I don't think I can go that far.
Another question. Thank you, Frédérique, for the nice talk. A part of your work is based on the classification of species as native and non-native. So I have a very naive question.
Oh, it will not be naive, I'm sure.
To do this classification and what are your conclusion?
So this is a very good question because in all my talk I made this dichotomy native introduced. But actually what is hidden behind these terms of non-native, at least in marine biological invasion studies, are two categories of species. What is really non-native that we know had been recently introduced, and what we call cryptogenic species. And cryptogenic species are a huge set of marine species from which we don't know actually what could have been their natural distribution range, because they are linked to this kind of habitats and this kind of activities or interface with human activities for a very long time. They are quite often cosmopolitan, but being true species. I mean, really true species. And so actually, no, it's complicated. And when we don't know, we put this name cryptogenic. This is a cryptogenic species, usually show the exact same pattern that the introduced species.
Other questions?
In your second story on algae, when you showed your map, I couldn't help to wonder what's going on in Roscoff. It seems like there's lots of admixture there. So I hope it's not a dirty lab leak story. Can you show us on the map? I was wondering because
there is not only Roscoff, actually.
Okay.
I wanted to make, I fully agree, I make it simple, but it's never simple.
So seriously, do you know?
Yeah.
Is it really
Ils ont du mal à avancer, hein?
Because it was a bit similar to the farm.
Yeah, but I have the answer for that.
Okay.
So I take this one, but it's the same. So yes, you're right. Actually, in Roscoff, we had, as you can see here, we have marina and 2 rocky reefs. And in Roscoff, we also have 1 farm. And so, yes, in Roscoff, you're right. One of the natural populations, which is actually established really nearby the farm, show, the part of their membership is to the genetic cluster, which are best explaining the farm. So you are perfectly right. And it's also true in Saint-Malo, not with the farm, but it's also true in Saint-Malo that there's something also a little bit different. But clearly, in Moscow, it's different because of the farming activity, which is located nearby. And you can see that the two samples from the farms, which are here and here, are very similar, but actually because they are exchanging seedlings.
Okay, but more generally, all this area shouldn't interpret that as that there's more mixture going on. This sort of colors in this structure.
You mean here or here?
Yeah, all this area.
Oh, yeah, okay, but all this area is very particular because the marina is a newly built marina that I was showing before. So this is ongoing. So that's why, and so admixed for sure, because there are influence of different things. But this work was made with samples, not including the one that I showed in this slide. So this marina where there is a lot of admixture is this one, the newly built marina. And actually, when we did the genetic analysis, we observed something quite interesting, that in February, in 2014 and up to, if I remember well, the third generation 2015, there was a lot of admixture. So a lot of diversity, genetic diversity, and we could trace back to different locations, source locations.
So the marina was just filled in with.
Absolutely. But what is really crazy is that, I think exciting for us, was that in the next generation, it stabilized the pattern completely and with no more influence from the different source. So I interpreted that as a kind of intraspecific priority effect. Importantly, the species is sulfur.
You say that this kind of species, you have a lot of admixture in reports, but what do you have evidence that genetic admixture has a positive role in terms of invasion?
I didn't say that.
Adaptative admixture, for instance. But do you have any evidence that...
I did not tell that I can prove for the relationship between the genetic diversity and the success or the status. I just said that there is strong propagule pressure that we know from different work that it contributes to the success of the establishment. And the correlate of that is that we observe large genetic diversity. And this large genetic diversity may be a fuel for selection on standing genetic variation. There are a few evidences. For example, there is a very nice study by a colleague, Caroline Teppold, on the green crab, showing that the invasion along the western coast of US is associated with a selection, positive selection on an inversion, which was part of the standing genetic variation, and within population which were transferred from the introduction area on the eastern coast. So, which was very, very highly genetically diverse, and in which this particular pattern, chromosomic pattern, was found. I don't say this is a rule. This is a really important question that we are fighting to address properly, I think.
This is something that you read everywhere.
Yeah, I agree.
With the risk or the success of invasion.
There is a set of, there are not so many examples. I fully agree with you, Arnaud. I fully agree. But there are some evidence in some cases, some example in which it works, and it was shown that there is this correlation. And there is the case of the adaptive integration is typically something at the between species level. Also, oh, I had something in mind. Sorry.
Continuing on that, do you have a way to test this experimentally, this progression between species? Like,
Integration on admixture and propagule pressure and high genetic.
The progression that you showed in the two species, can you experimentally show the effect on the...
I hope so. I hope so that we will be able to investigate more the functional part of it.
So you showed that there was spillover from the port population, natural population. But what is the evidence of the impact of those spillovers on the natural community? We see CSE impact on variable environmental.
For this seaweed or in general? Because in general, I don't know in general. For this seaweed, there is little impact actually, but and in general, I guess it's really dependent on the interaction within the community and what are the niches that are empty or likely to be. So I think it's difficult to have a general question answer to this question.
Yes, report or something like that.
Yeah.
You want to comment on the activity?
No.
So maybe we have one small question online and then we go back to the room.
Yeah, we have a short question online. It's more about the biology of the species that you studied. The person is asking whether you think that given their temperature range, it could also be introduced in tropical waters.
No. The Robusta species is from warm temperate areas, and the native one is from cold temperate areas.
Yeah, just to go on with what Philip was saying, we have good examples, for example, in freshwater.
Yes.
There have been massive impact, for example, fungal mussels and
Okay, but impact. Okay.
ground mussels on local communities, on the dynamics of diversity, but also on the systems.
No.
What about? Are they...
Just because I think there is a misunderstanding. I understood the question of Ophélie regarding the spillover from ports. So the direct connection between spillover from ports and impact on community, it's not well worked as a question. But to evidence that marine introduced species have impact on community and ecosystem levels, there are a huge number of evidence and case studies. And so perhaps this is the answer you expected, not talking about only spillover. And in that case, yes, we have, of course, all the story about sleeper limpet, Cropidula fornicata really changed massively the habitats, really changed every component of the abiotic environment and then the community. I have Sargassum muticum to worldwide level. We have now blue crab. So there are many examples of massive impact. How long it lasts, it's something else.
Maybe one last question.
Is there any evidence that you have natural populations which become maladaptive because of gene flow between this one?
I don't have evidence, but this is definitely a very interesting question. I don't have evidence in mind. But yes, it's a very important question. This is, I did not talk at all about this aspect, but the port environments are really particular, exerting strong selective pressure. So you might imagine that at some point, some specific adaptations to this environment evolve. And so for native species inhabiting both port environment and natural habitats, it's quite obvious that you may imagine that some kind of maladaptation might happen in native populations. So it opens important questions regarding the use of these habitats as nurseries or refuges or biodiversity, etc., which are ideas that are now proposed.
Thank you. So maybe please join me in thank you, Fred. So please stay with us with the apéro that it would be just outside. There are drinks and snacks for everyone.